Hello, everybody. Thanks for coming. I'm Julie Passarelli. I'm the exhibits curator here at Cabrini Marine Aquarium, and it's my pleasure and honor to introduce tonight's speaker, Dr. Steve Weisberg. Dr. Weisberg is the executive director of the Southern California Coastal Water Research Project Authority, also known as SQUIRP, which is a research consortium formed by 14 leading water quality agencies in California. Dr. Weisberg received his undergraduate degree from the University of Michigan and his PhD from the University of Delaware. His research emphasis is on developing molecular assessment tools to support environmental monitoring programs. He also focuses on linking the needs of the management community with science. He serves on numerous advisory committees, such as the State of California's Clean Beach Task Force, the California Ocean Protection Council Science Advisory Team, the California Sea Grant Program Advisory Council, and the California Water Quality Monitoring Council. The title of tonight's talk is Advances in Beach Water Quality Monitoring. Please help me welcome Dr. Steve Weisberg.
What a pleasure to be here. We've had such a great relationship between our organization, SQUIRP, and the Cabrini Marine Aquarium. You all run such a class operation here. We've been very fortunate to have held a number of our scientific events here, and so it's an incredible pleasure to get to meet some of the people who are the supporters of this program, and then hopefully share some of the science that we're doing that might be relevant to you. I'm actually a little bit intimidated because I saw the list of speakers that you have in your program. An incredible bunch. Julia, congratulations on a wonderful set of speakers you've been able to accumulate. And I understand I'm following from somebody who talked about copulation of animals in the ocean. So I got a tough job in front of me trying to keep this entertaining. But fortunately, I'm working on beach-related issues, and that usually is enough to keep people interested because beaches are such a large part of our culture here in California. And in particular, I'm excited because this is an exciting time to be a scientist. We're really in a transition period of how we do science. The opportunity to move from some of the things that we have done historically to genetic methods, doing things by measuring genes, just advances where we can go. And that's what I'm going to be talking to you about today. So let's jump in. The starting point is something I probably don't need to tell you about because you parked across the street or across the parking lot from a beautiful beach. Beaches are a large part of our culture here in California. It's the number of visitors we have is very large. The economic value is very large. Because of that, California has the most robust beach monitoring program in the nation. About a third of the total beach monitoring in the country, including Hawaii and Florida and all those other places, is done here in California. We have state regulations that were put in 1998 that require all beaches with large numbers of swimmers to be monitored at least once a week. In the LA area, most of the beaches are monitored daily. It's kind of a big deal, but it's even better than that. In California, we require that the data be made available. And we require that when a problem is found at a beach, that signs are posted on the beach. In many states, it's not the case. They collect the data, they use it for things, but they don't notify the public. We do a wonderful job of that. And we do it in 3 or 4 different levels of ways. So there's what is posted on the beach that day with a sign that might look like that, what gets posted on a website, and most of the counties, LA being one of them, maintains very active websites. But there's also, I'm hoping that many of you are familiar with, or if you haven't become familiar with, Heal the Bays report card. Because what they do is they assemble the data and look at the beaches over the course of a year or over the course of a season. They actually grade the beaches from A to F. So it's a real opportunity for you to know not just what's happening on the beach that day, but to also know how the beaches are doing in general. In fact, one of the biggest things that I run into is that people get a little bit on the concern side because, oh, look at this, I heard about this beach and this beach getting a warning sign. I'm here to tell you that the beaches we have here in California are among the cleanest in the nation. The reason you hear about it is because we do such a good job of monitoring it and finding out about it. We have a very small number of beaches that have a problem. Most of them have a problem very much like the beach here, have a very low-level problem, infrequent level problem. And most importantly, we know where those beaches are, and there are active cleanup efforts around us associated with each of them. And I'll even tell you a little bit more about what's going on at your beach right in front here later today. But, there's a but here. The methods that we're using to measure at beaches are 100 years old. Now, that doesn't mean that they're bad. It just means that they're 100 years old. So the way that we typically monitor on beaches, actually, can you all hear me without the microphone, or do you need me to stay close? Mike's better. Mike's better? Okay, then better without, even better. All right, so the methods that we use are what are called culture-based methods. What we do, the typical way we go about doing it is we grab a sample of water, usually in a 100 milliliter bottle, about that big. We take it, we filter it, we put it on filter paper. We take the filter paper, we put it in a growth medium called an agar, and then we put it in the oven for, depending on what test we're doing, somewhere around 24 hours, sometimes 72 hours. And we come back, we pull out the plate, and we measure the little dots, the bacterial colonies that are growing on there. And theoretically, what you've got is if you put it on a special type of growth media and you bake it at a certain temperature, you'll get the target species that you're looking for. And we've been doing that for a long time. And it works, and it's very repeatable, it's very doable. Again, there's always the but. The but is you let it sit in the oven for a day or two. And so the sign that says bacteria levels exceed limits for safe swimming really should say, it was bad yesterday, we don't know about today, come back tomorrow, we'll tell you about yesterday. What we're really saying to you is, you shouldn't have been swimming here 2 days ago, you're going to get sick. So if you're looking at it from a water quality management perspective, and you're kind of looking at the long-term average of how that beach is doing, fantastic. But if you're wanting to know what's happening now, not so great. And so this is where the real advances are taking place. The solution is moving from culture-based methods, which take, in the shortest of a full day, and up to 4 days, depending on what test you're doing, we can do in 2 hours by not measuring the bacteria themselves, but measuring the genetic material that's there. So how does that work? Well, we go about collecting the water in the same way in small little buckets. We filter it again, put it on filter paper. But then what we do is we digest the filter and we extract the DNA. And what we do with the DNA is you essentially make copies of it. When you make copies of it, what you're doing is you're grabbing essentially a DNA slice that you have created that matches the DNA of your target. And by using a series of heating and cooling processes, you cause the DNA, the double-stranded thing, to separate. And then you cool it, and it reconvenes. But when it does, it doesn't do it with its original strand. It does it now with this target that you've put in there. Right? And when it does that, what we do is we put what we call fluorophores on that. And so when it attaches, it releases this fluorophore, which is light. And so what we're able to do is measure the light that's released. And if there's a lot of that bacteria in there, then you'll get a lot of light. If there's only a little bit of bacteria, then what you do is you repeat this sequence of cooling or heating and cooling many times. And every time you go through a heat and cooling cycle, you're doubling the amount of bacteria and doubling the amount of light. And so what happens is, instead of measuring the light itself, we're actually measuring this curve of how many heating and cooling cycles it takes before we get light that's measurable by our device. And if you start with a lot of bacteria there, it doesn't take long to get to the point of having measurable light. If you have very little bacteria, it takes a long time to get there. So it's a very simple process. Again, if you've got a PhD in microbiology, but it's very doable, and we have the technology to do this. This type of technology is really what's being used in the hospital industry right now. So if you want to go measure things in the hospital, we're doing more and more like this. And you'll hear it called quantitative polymerase chain reaction. So the polymerase chain reaction is the number of times you're breaking this and reforming it. So what I'm going to talk to you about the rest of the day is what this does for you. So first thing, as scientists, we can develop these methods. But that's really the start of the process. What happens after that is we need to prove that the methods really do work. So we go through a series of steps. First step we do is we take the old method, EPA 1600, that's the number of the method that's with filter paper and counting things. With this qPCR method, we do a correlation. So we get the same answer between the two? The answer is not bad. As a correlation, that's not bad. In fact, we have a lot more noise at the low end. Doesn't really bother us. We're down near the low end of the detection limit because you're really not interested in those places. Those are the places that don't have much. You're more interested in what happens out there, and the correlation gets even better. But what's fun about this is that you say, oh, well, it's not a perfect correlation. Maybe this new method's not so great. Well, what it does is it forces us to think about it from the other direction. Well, maybe the old method's not so great. And one of the problems that we find with the old method is you're putting things on a growth media. When you put it on a growth media, it's not like it's going to grow exactly what you want it to grow. It's going to grow anything that grows on that growth media at that temperature. It's not as specific. And so what we do when we're doing the measurements on the beach, we're not actually measuring pathogens themselves. I'll come back to that later. Pathogens themselves are very difficult to measure. The problem with pathogens, the biggest problem with pathogens, is beyond the measurement challenge associated with them, is that they occur at very low densities. So you'd have to get a good sample, you have to get like a 55-gallon drum of water and filter it to find the virus. What we use instead are bacteria that occur in your gut that are indicative of fecal material, and they co-occur with the pathogens. So if you're not getting any of these bacteria there, then you wouldn't be getting the pathogens. All right? And the indicators that are most common in your gut are these two species, Enterococcus faecalis and Enterococcus faecium. All of these others are ones that grow in the environment. You really don't want them. And what happens is, though, they're too close to the targets we're interested in. And so what happens is you can see that in a normal sample, only about half of what we grow is the thing we're truly interested in. When we do the molecular methods, the genetic methods, we can target to exactly the species we're interested in. So the fact that that correlation between the two wasn't great didn't discourage us. It actually encouraged us. So the next step that we did as scientists is what we think of as the gold standard. We actually do epidemiology studies. With an epidemiology study, what you do is you go out and you're asking the question, are people getting sick? Are we able to correlate the measurements we're taking with rate of illness? So the way we do those kind of studies is we go out and we get people at the beach and we enroll them in the study. So we kind of quiz them, and what we're doing is we're looking for people who haven't swam for a week before the study and don't plan to swim for a week after the study. And then what we do is we watch where they swim and we sample where they swim to look at the bacterial concentrations at those locations. And then we call them about 10 days later and we give them a list of symptoms to watch for and we say, did you get sick? It's very complicated in how we do it, but in a sense it's very simple. It's a correlation between did you get sick, and we've seen a lot of indicator bacteria. And we've done these studies where we use both the old culture method and the new genetic method. And what we're finding is that in almost every case, the genetic, what we call molecular methods, outperforms the illness rate, or the illness rate prediction. So in a sense, we have a tool that's faster, it's better because it's better correlated, it also turns out it's cheaper. So, you always heard about faster, better, and cheaper. You give me any two of the three. This is an opportunity where we're actually getting 3 out of the 3. But as scientists, we don't stop there. So it's great that somebody with a PhD working in a research lab can do it. How transferable is it? So I'm here to tell you, this is something where we're actually fairly far along with. We at my organization, Squirp, have trained 14 laboratories in California in how to do this methodology. And it's really worked well. What we did is we brought them in, we did a three-day training workshop, hands-on, how to do it. Then we sent them back to play with it in their own lab, and we went over there to kind of observe them. And then we did an intercalibration exercise where we send everybody the same sample, we process the sample, and we're looking to see how close is everybody. And the answer is incredibly good. We gave people 3 different samples, one at high concentration, one at medium concentration, one at low concentration. What you're seeing here is the variability that we'll see within a normal laboratory that's doing it by itself. And what you can see is, look how close the laboratories are. That variability is in fact tighter, less variability, than we see with the old method. 14 labs can do it as well as we can do it. So it's very transferable. Okay, so I'm giving you a great success story. Isn't this wonderful? Look at all these things we can do. We can transfer technology. We start to transfer it. People start to use it. What happens? We realize our next biggest challenge is, wait a minute, I can do it in the lab in 2 hours, but this is Southern California. How much traffic is there? Right? And so what we're finding is that all the laboratories would go out, they'd send their people out to sample, leave at 7 o'clock. It would take them 4 hours with California traffic to go to the beach, collect samples, bring it back. Then they bring it back, oh, wait a minute, it's lunchtime, I'll process them after lunch. We weren't getting answers till mid-afternoon because of that. And then you got to go find the health officer to get the information by the time you find them, and then send somebody back to the beach to put the sign up. It's 3 o'clock. Didn't do any good. I mean, it's better than having the answer the next morning, but really from a swimmer's perspective, how much better? I'm done with my day at beach at 3 o'clock. So great in theory, less in practice. So, challenge to the scientists, what do we do next? What we're in the middle of doing right now, and this is where it really just gets fun, we're at the point of miniaturizing it and putting it in a device that's small enough to take in the field, and we're making it simple enough that a lifeguard could do it. So the idea is we're developing a device, and there's a prototype, a working prototype. The device on the right, the grayish one, is the thing that we pour the water into, and it digests the sample and pulls out the bacteria. The thing on the left, the yellow thing, is a miniaturized version of the thermocycler, the thing that changes the temperature, and it's all automated. Now, there's a long way to go from that prototype to actually having it in the field, but that's where we as scientists are going. Getting it to that point, we have working prototypes. So it's just neat to see how we're improving that technology. And it even gets one step better. Having it field portable starts to open up new possibilities. So right now, when we do the beach monitoring, it's a problem. What do? You put a sign up and say, please don't swim here. But really what you want to do is fix the problem. If you have a device in the field and it gives 2-hour turnaround, now all of a sudden what you can do is you can start walking the beach. And while it takes 2 hours, that portable device can run a new sample about every 3 minutes. So you take a sample here, you walk down the beach, you take another sample, walk down the beach, take another sample, and you go, and then you walk in the other direction, take a sample here. And all of a sudden you can start to see where is the contamination coming from. And then if you find something like this, you can start walking up and taking samples there. So our ability to go trace the samples goes way up. So again, the science of faster methods is giving us new applications that will help us improve the beach water quality. We're also getting to the point of developing an automated system. So in a sense, if all I got to do is have the human pour the water in, well, wait a minute, why don't I just take a pump and pump it up into the system? And we've got a prototype of that too. Here's basically a 55-gallon drum that includes, again, something that looks like this. It's in prototype form, but we've actually used it in 3 different locations already, and it works. Is it stable? Is it manufacturing? We're still a few years from that. But because we don't have to do that culturing, all right? We have to culture it, forget the timing, just how do you create the incubators and all those kind of things to do that? You can't do it. But this, you can actually put out in the field. So wouldn't it be wonderful, instead of even having a lifeguard process a sample and say what it was 2 hours ago, now you have samples that are running throughout the night. Before you leave for the beach, you go to the website and you look, what was it like that day? What was it all night? What's even nicer about this is what we do right now with our sampling is we go sample a point in time. Well, we have high tides and we have low tides. And maybe you're there at a time when the water's moved in this direction. Well, if you're doing it continuously, and you're getting a sample in a sense every hour, all of a sudden that's not a big deal. You're getting to see what it looks like. Do I want to go to that beach? We're getting really close to being able to do that. So, kind of fun. But it gets even better. I refer to these devices as Trojan horse. People want them in their laboratory. They want to be able to do these genetic methods so that they can do it faster. But once you're doing genetic methods, wait a minute, there's all this other, in a sense, hidden information that's in there that opens up new worlds of possibilities. So it turns out that the genetic markers that go with human poop are actually different than some of the genetic markers that go with dogs or cows or other kind of things. And we've been able to identify what are those genetic sequences that differentiate them. And we've done a number of studies, a bunch of blind studies that allow us to define what's happening there. And this opens up a new world of possibilities. Because if you want to clean the beach, wouldn't you want to know, is this coming from human fecal material? So in which case, go look for a leaky sewer pipe, okay? Or maybe a baby's butt that, that was in the water. Or is it coming from birds? In which case, I put there, should I hire a falconer? There are a number of groups that have found birds to be the problem, and they literally hire a falconer. The falconer doesn't actually go and kill the birds, just flies around and all the birds run away. And you know what? Beaches get cleaned up that way. But if it's a human fecal source, don't bother hiring a falconer. So all of a sudden, managers, when they have this, just have an incredible ability to do it. And we are at the point where we can absolutely do this. We now have a manual on how to do this. And these are all the sources, human, dog, cow, horse, pig, chicken, deer, and gull, that we have great confidence. When I say great confidence, like 95% sure when the signal says it's a gull, it truly is a gull. And where it's off a little bit, there are some, for instance, some of the markers, the dog and the cow, as for instance, might cross-react a little bit. We know that. So we really have the ability to separate out the human-caused beaches from the dog beaches, from the cow beaches. And when you start to work upstream, this is where it gets to be really helpful. Because let's say you find it's coming from a particular stream. Well, what happens when you have a big horse farm on that stream? What happens when you might have septic systems on that stream? What happens, when you might have birds, now all of a sudden we have the ability to start saying, where is it we want to focus our investigative efforts? So new tools because we can use these genetic methods. It gets better. It opens up a whole new set of interesting management questions. Like, does it matter in my warning system whether it came from a human or a dog or a cow? Is one more pathogenic than the other? Because if it is, maybe it's less important to clean one type of beach than another. Or maybe these indicator bacteria, Enterococcus, the things I was mentioning before, maybe I should use a different threshold for each of those beaches. Well, it turns out EPA has opened the door for this through something called quantitative microbial risk assessment. And the reason they've done this, and the reason it's so important, is it does matter whether or not the fecal source was human or something else. It matters a lot. So the reason that we measure these Enterococcus is because they occur in the human gut. And in the human gut, you're going to get a lot of pathogens. So if you are sick with something and you poop it out, it's going to be of concern to other humans. But do birds have the same pathogens? Do cows have the same pathogens? So we've done studies, and I say we, I mean scientists as a whole, to look at what is the relative probability of illness depending upon the different types of source. So you can see that if you go over here and look at raw sewage, if you have Enterococcus values at 35, which is one of our thresholds that we use for whether or not a beach is a problem, this is the level of illness we expect. Interestingly enough, if you go to secondary effluent, it's even higher. Interestingly enough, Don't swim where the cattle are, because it's pretty darn high. But if it's birds, look how much lower. This is a scale that's a log scale. So basically, it's 100 times safer. If you have an enterococcus failure, it's 100 times safer to swim at a beach where it has gull poop than has human poop or has cow poop. So again, I want to be careful here not to say ignore gull poop. One of the things that somebody asked me one time is, so does this mean I shouldn't worry about gull poop? Okay. If any of you have small kids and a gull were to poop in the bathtub, would you put your kid in the bathtub? No, you'd clean the darn thing out before you put him in there. But if you had a choice between putting him in a bathtub that had human poop or gull poop, put him in the one with gull poop. So, but from a management perspective, see, I'm starting to catch up to the copulation people. But the fun thing about this is it now starts to open up interesting management questions. Like, do you really want to do something? Do I want to put as much money into cleaning up the beaches that have bird poop as I do the ones that have human poop? And where do we put our restoration efforts? So, as I mentioned, EPA has opened the door to setting up what we call site-specific objectives. In other words, I might allow a higher number for this enterococcus measurement, this correlative indicator, if the fecal material is not human. And as part of this quantitative microbial risk assessment process, you have 3 major steps. Step number 1, source identification. You got to have those tools to say, you got to be sure that it's non-human, because we don't want to lower a standard when there's a human material there. So you got to be darn sure about it. And so again, this is where all those tools for separating human and non-human come in. The second thing you need to do is, in essence, what we're saying is that when we set up the original relationship between this enterococcus fecal indicator and pathogens, we did it at human beaches. And maybe at these new beaches, we need to now set up a new relationship between the indicators and the pathogens. But in order to do that, you need to be measuring pathogens at the beach so that you can create that relationship, figure out what is the right threshold. And then you do a risk model to identify that. Interestingly enough, again, this is where the advance in science, the ability to use these molecular methods comes in. Because if you had said to me, Steve, 10 years ago, well, let's just do the pathogen measurements, I'd say, how? Pathogen measurements are a real pain. Pathogen measurements, okay, are really difficult because many of them are what we call obligate anaerobes. They will only grow in an environment that has no air. All right? Basically, they're things that kind of grow in your gut. All right? And if you put them in a regular laboratory that has air, they won't grow. So you have to create this incredible laboratory circumstance. Very, very expensive, very difficult to set up. Using genetic methods, I'm not growing it. I'm just measuring the DNA. I don't have to have that special circumstance. Second problem, their growth presents a health risk. Do you really want to be growing those pathogens? Do you want to be the one working in the laboratory? We have specialized laboratories to do it. You've seen the people in the blue suits. That's what you got to do to measure some of these pathogens. But if all you're measuring is the genetic material, don't worry about that, because it's not live pathogen. You're just looking at the DNA. Pathogen growth can take 30 days. It takes a long time to grow those things out. Besides the expense, look at all the time that's involved. Again, when we do the genetic methods, it's the same 2 hours. All we're doing is looking at how do we match up a genetic sequence. It doesn't matter whether it's a genetic sequence for the enterococcus indicator or for the fecal source marker or for the pathogen. It's the same basic process. So the development of the tool, the thing that allowed us to do it faster, now allows us to investigate things at a whole new level. We can look at the pathogens. So, I will tell you, right across the street there, right across the parking lot, is one of the places where this quantitative microbial risk assessment is being done. It is one of the few places in the nation. It is one of the pilot projects at the national level for how to go about doing this. Why? Because there's been a lot of suggestion that the biggest problem that you have at that beach is not a fecal human problem, but it's largely a bird problem. So it might be a candidate for exactly that. Maybe you want to reinterpret the numbers that you're seeing at that beach. So this is a pilot project. It's a joint project between my organization, EPA National, and a few other organizations to look at this. So, as a history, many of you know, there have been multiple approaches, a lot of money. I mean, literally, last I looked, it was in excess of $20 million has been spent by the ports and some of the local organizations to try and clean the beaches. We've done things like look at all the piping systems and rebuild a lot of the piping systems so that we're sure that there isn't any leakage. We've done sand replacement. One of the things we found at other beaches is that sometimes it's not the water that contains all of the bacteria, it's the sand. Sometimes these enterococcus just regrow in the sand. And so one of the things that you all have done locally is sand replacement. And you all have been here enough to see some of these things. Remember the groin? One of the things that was, a big thought was, oh, well, if all we do is create a little bit more circulation, we'll solve the problem. Pretty expensive fix. And I'm sure that many of you have seen the bird covers, the kind of wires that go over the beach to try and keep the birds out. You still have a problem. Okay? Things like these have helped. It's certainly better than it was, but it's not perfect yet. So, one of the things we've been doing is initiating this study, which we initiated last year, and you'll see a lot of our samplers out there. So the kind of things that we're doing, the first stage of the QMRA risk assessment is to rule out the human contamination source. So towards that end, we've done a lot of studies in the water, in the sand. You'll see some of the eelgrass and the rack that comes up. These indicators will grow on that. Trying to understand the patterns of those things. We've done over a wide range of conditions. We've done 36-hour studies, so we can look at different tidal conditions, different time of day conditions, day of week. One of the things we're really curious about is, do we see a bigger number when there are more people in the water? In other words, is it a reflection of what we sometimes refer to as butt soup? People putting their kids as little tea bags in the water. And we measure both the indicators and the source markers. We also are doing work to look at cameras into the piping infrastructure to see if we can find things. So, these are the sites that we focused on. This one called CBO2 is in fact where most of your beach monitoring for that site is. So when the county puts up a sign, that's the site. These are the other sites, and we even have a site on the outer. So the summary of what we found so far. First, you don't have a terrible beach, but you do have an imperfect beach. So the standard that we use in the state of California for instantaneous measurements is 104 per 100 milliliters of water for this thing we call enterococcus, this indicated bacteria. And what these are, box and whisker plots. This is the median value. This is the 25th and 75th percentile values. These are the 5% values, and then those are some of the outliers. And so you can see, I think I have here we go, the single sample standard. And so what you're seeing is that particularly for site 3, that's kind of in the middle of the beach there, in 2016 with gobs and gobs of samples, we probably had about 700 samples. About 25% of them for that site, similarly for that site, similarly for that site, were above this 100. Now, we don't have anything that's really sky high. I'm at the really nasty beaches. If you've got a lot of human poop on those beaches, you'll be sitting up, 10,000, 100,000. We're not close to that. We never got a sample anywhere near that. So you have kind of one of those beaches that's kind of on the margin. But from a regulatory standpoint, from a is this a clean beach perspective, kind of giving you the opposite. 25% of the time, you're above that single sample standard. So this is a beach that you'll want to clean up further. And it's a good candidate for maybe what we're seeing is just some residual from things like birds. So going to the birds thing. Bullet number 2. Birds are clearly a major source. And I'm going to show you some data in a minute. We see it in almost every single sample we collect. And the spatial and temporal relationships match enterococcus. In contrast, the human marker we rarely find at your beach. And it shows no pattern related to this enterococcus. So, birds. This is the correlation between enterococcus, the indicator bacteria, and the gull markers. and this is in water, you can't get much better relationship. Biologists, we pray for relationships like this. They're always fuzzy things. They always look like, Jackson Pollock paintings. It's almost never that clean. And even in the sand itself, we find almost the same relationship. Let me show you what happens with humans. So again, the enterococcus, you see a little bit, most at site 3 and 4 in the middle. That's what the birds look like. That's why the correlation is so high. I don't even bother showing you the correlation for human, because that's what it looks like. First off, almost everything is low. Remember I said this is the 25th, we can't even show you, the lower values because they're all zeros. We get a small percentage and there's no pattern that relates back to the enterococcus. So birds appear to be a big problem at this beach. So I'm not giving you a final answer here. I'm illustrating for you how science has advanced to the point that we can kind of figure out where we go next. There's kind of a path forward for us to maybe consider that this is a beach where you might think about a different standard for the beach if it's all gold material. Next steps, the human fecal signal was not zero. We want to be very conservative in applying this QMRA. We do not want to say, oh, let this beach go. Lower the standard, just let it go. We have to be very certain. And so the people, essentially the managers of the beach, it's not the scientists, this is the managers of the beach, going, we want to be really certain. So what we're going to do is we're going to continue to examine the infrastructure, consider to do more piping, see what we can fix there. We haven't found any relationship, but everybody wants to be sure there's no leaking infrastructure. The other thing that we're doing, and if you had been out on the beach literally today, we collected hundreds of samples on the beach and in boats that go out past the beach, looking to see if there's anything that's coming in from the harbor towards the beach. Again, another possibility. If that's what's happening, if there's some human fecal signature that's coming in from that direction, again, you don't want to change the standard for the beach. So these are some of the things that we're doing. Once some of those things are done, we'll pursue either later this summer or next year some additional source identification. So again, an example of where the technology has just exploded in terms of enhancing our ability to manage, to understand the sources and manage the beaches in a whole different way. And it's happening at your beach. So I've talked about what molecular methods do for you in the context of beaches. What I'd like to do is add to that a little bit by saying this technology doesn't apply just to beaches. It is opening the door to what we do in all kinds of biological management. I'm showing you, I'm a little bit older than some of the people in this audience, although there are a few who look older than me. You might remember it, this kind of progression. When I was in high school, I graduated high school in '71. We're still using slide rules. When I got to college, I got my first handheld calculator. It had one memory. Okay, I put one thing in the memory, and it cost me like $200, would probably be equivalent of like $1,000 now, to have that little thing. By the time I went to college, you had these TIs that had all these functions in them. And about that time, we're starting to get these big computers. They were an entire room. And anybody remember punch cards? Yeah. Okay. And you remember actually falling and watching all the punch cards go out of order, or running a 2,000 punch card thing, and one got stuck, and you couldn't figure out which one it was, and your whole program blew up. This is where we were in the late 70s. 1984, IBM came out with the first PC, right? No hard drive, just floppy drives. You want to run Word perfect? You didn't have Word back then. You actually ran it off the floppy drives, and you had to put floppy in and put another floppy in. That's where we were. But you can see how quickly this was advancing. And of course, I'm betting everybody in the room has one of these. There's more computing power in this than there was in this in the 1990s. So that's how fast it's going. For my generation, or for the next generation, I'm kind of at the end, genomics is our equivalent. If you kind of think about it, we didn't even know what DNA was until the 50s. I mean, we're kind of thinking about it as a possibility. What would, the whole double helix thing, that was 1953. That's not that long ago. The first time we ever did a genome sequence was 1976. The first time that we were able to get those markers, it wasn't until 1994. That's like 20 years ago. All right? DNA-based measurements is now the focal point of medical work. All right? You get an HIV test, how do you do it? They're not culturing that stuff. It's all done the same way that we're talking about here. Anti-terrorism, how is it done? There are actually drones that fly. I told you about how we're miniaturizing all this stuff. They got a lot more money than we do. Okay? They're miniaturizing it a whole nother level. We're basically just borrowing the technologies that they've already been developing. But essentially in warfare, what they're doing is they're flying over to look for kind of those biological weapons, to see whether or not there's anything being released on the battlefield. They have that technology. And of course, everybody sees CSI and all its offshoots. It's just now beginning to affect the environmental field. For beaches, we were really the cutting edge of this stuff. But it's not just beaches. So let me give you another example of where this is happening. Harmful algal blooms. I'm assuming most of you have heard of harmful algal blooms and aware of the number of harmful algal blooms. The way that we used to do it, the way we still predominantly do it, you go out, you collect big bottles of water, you take them back, you put them on a slide, and you take some poor schmo, and you let him sit there and go blind. And do it for hours and hours in a day. And it works. This particular guy right here, I know him well, he's 6 months behind in any sample you give him. Okay? Because it just takes that long to do. Here's how we're starting to do it now. Again, we have working prototypes of this. Okay? You put a mooring out there, you pump things through. We have what's called the gene microarray. It's similar to the kind of things I was just showing you, but basically it has multiple species as dots on that thing. And if that species is there, the dot will show up. We actually then have a camera that watches those dots and produces data in real time back to the web. Automated, continuous. All right? Don't have them everywhere. Again, we're still at the prototype stage, but we have working prototypes. I think for this particular device, there's probably about a dozen of those prototypes now being used around the country. So are we going to get there with beach bacteria? The people that I'm working with to develop the beach bacteria thing are the same people who are already doing this. In fact, the only difference is this kind of microarray is a little bit easier because it's more of a presence-absence thing. You just have to see, do I get any of that gene there? What we're trying to do is actually quantify. So we have to do more of the multiple steps. So it's a little bit more, in a sense, advanced technology. But it's the same basic underlying process. What are the other opportunities? Well, I started out by talking about human health monitoring on beaches, but it's not just beaches. Harmful algal blooms, I just mentioned. What about seafood safety? Okay? It's the same thing. You worry about things like vibrios. That's not what we're worried about on the beaches, but you sure don't want to be eating vibrios that occur in an oyster. The same technology allows us to measure what's going on in an oyster. It's not just the human health part. It's the environmental quality assessment. Okay? What we're doing right now, there are a lot of fisheries monitoring programs that are out there. And what do? We drag nets. Right? It turns out that fish are constantly shedding scales and other kinds of mucus, and every bit of that contains genetic material. What we're doing now, what we're developing the technology for now, is scoop the water and just grab the genetic material right out of the water. Don't even have to grab the critters themselves. Okay? You can grab the critters themselves, and you can do the genetic work. But we call it extracellular DNA. And we can actually measure. We can do a fish survey right now and tell you what fish species swam through that water in the last period of time. Okay? It's really incredible to watch. It is all developing. I mean, it's developing almost like by the hour. Nobody was doing that 5 years ago. But we've gotten to that point. The technology is really just developing. We have microarrays to basically look at, are the organisms. We can look within the organisms to see what genetic pathways are being what we call upregulated and being downregulated. What chemicals cause those pathways to go up? In other words, when they get exposed, what biogeochemical pathways are going up and what genetic patterns are going up if we can find those markers. So in a sense, If you have some heart trouble, you might look for blood enzymes for what's going on in your heart. The fish do the same thing. So we can look inside the fish and look at what are the genetic pathways that are going up and down to identify what kinds of things they've been exposed to. Just all kinds of fun. Species management. Invasive species. This is a big port area, that's why I chose that example. Somebody comes in. You want to know what happens. Do you have in the ballast water? Do you have invasive species in that before you let them discharge the ballast water? Again, same thing. We can look at the genetic material. We can do it in a series of hours. So again, it's being done more at the research level still. We have to develop all the genetic information for all the different species we're interested in, but we're exactly doing that. And then endangered species monitoring. I just went out to a seafood restaurant. Thank you all very much for taking me out to a seafood restaurant. I ordered ahi. I could actually, it won't be long before I got a tricorder that I take to the restaurant, stick it into the fish, and look to see whether or not it's truly ahi. And there have been exactly those kind of things done at fish markets and at restaurants. There were some student projects done in New York where they went around and actually used this technology to assess, do they have the right species? And it turns out about 20% of the time, they were not selling the things they were claiming to sell. So again, that's really at the marketing level, but it's really at the endangered species level. If you want to be looking to see, are endangered species being found? So I want to leave you with the idea, because they asked me to leave some time for questions, that you are on what we just talked about for beaches is already changing how we manage beaches, and molecular methods cause you to be on the cusp of huge changes in the future about how we do all of our environmental monitoring. So with that, I'd like to thank you, and I'd be very glad to answer any questions.
Give a few more lights here, Steve, and then we'll. Some questions. Is that a question right there? Right there.
Well, it was going to be a two-part question, but you already answered the second part. The first part was what depth and how close to the beach you monitor for the beach pathogens. And the second part was going to be, it seems like the nanotechnology and the advances that you already talked about, and GPS, remote monitoring, nano machines, doing these PCRs.
You're going to be able to have like a map on the internet that just tells you everything.
Okay, so what do we typically do right now? Is we sample at ankle depth. So you go into water about that deep. We do that for two reasons. One, I guess I'll just call safety, because you want to measure at different beaches, and there's some beaches that have crashing waves, and you don't want to be sending your samplers out of those beaches into deeper water, waist deep water, because they're going to get knocked over and it's a mess. But we also do it because when we've done those epidemiology studies, we've actually done measurements of where the greatest health risk is, and the greatest relationship is with the water that's really shallow. And so you want to go to the place, as you get deeper, in a sense, there's more dilution. And so you want to go to the place that has the highest number because you want to be on the safe side. At this beach, it probably wouldn't matter because you don't have a lot of tidal influence. But
I wonder how many people here tonight will go up tomorrow morning and go to the local beach and paddle in the water for 5 minutes. Can have a show of hands. It's pretty eye-opening.
well, again, one of the things I tried to emphasize at the beginning is that we have the best monitoring programs in the country in California. And the number of beaches that we have that have really high levels, not very many. And even this beach, okay, right in front, where we have these low levels. First off, my gut feeling, I hate to use the word gut because it's kind of a, but my gut feeling about this is that your biggest problem, again, I wouldn't be ready to regulate differently because the science isn't finished here, but my gut feeling is that you have a bird-oriented beach, and most of the problem, so when you're above that threshold, again, given the numbers, you need to be a lot higher before you truly have a health risk. But even if that was human fecal material that caused it, we're very conservative in California. That 104 number I told you about, what does that do in terms of illness? What does that mean in terms of illness? It doesn't mean you're going to die. First off, the illnesses that we're getting are what we call self-limiting diseases. You're going to like poop a little bit more. You're going to, not feel perfectly. You might even get a fever, but you're not going to die. All right? More importantly, when we do these epidemiology studies, the background concentration or the background rate of illness for things like diarrhea is about 3 people will get, out of 1,000 will have diarrhea over the next 2 weeks. Okay? So if you have human-related fecal material that caused you to be at 104, that number jumps to 6 out of 1,000. So in essence, you didn't get to the point of I'm going to be sick. You got to the point of going, I'm going to double my background. Okay? I have an extra 3 out of 1,000 chance. Okay? That's not that big. We have a very conservative, and with your particular beach, I don't think it's anywhere near that high because it's mostly bird-oriented. So there are some beaches that, I've been in California for 21 years and working on this. There are some beaches that used to be a lot worse. The beauty is with our monitoring programs, we've gotten very good at identifying those beaches. And actually, we are very fortunate to have with us today John Bishop, who is the, Chief Deputy Director of the State Water Board. John was one of the people who put forward a Clean Beach initiative out of the Water Board. They put about $100 million into beach cleanup. And in essence, a lot of the things that we're doing here, identifying which beaches have a human signature, not having a human signature, were used to figure out which beaches we need to go clean up. And the number of beaches we have that are in bad shape right now is very small in dry weather. Okay? If you ever go and look at the health report cards that come out of Heal the Bay, you'll notice a very big difference between wet and dry. We've done a very good job of cleaning up the dry. The wet, it overwhelms the system. That's why they tell you, don't go in the water for 3 days after. Again, you're not going to die, but the illness rates will go up in the winter.
Okay, thank you.
So,
Quick question. You mentioned the tides. Is there any correlation between low tide and high tide? You didn't mention whether there was. And you check for industrial runoff and things like that?
okay, so first question, repeating it in case you can't hear on the back, is do tides matter? Do they cause an effect on the counts at the beach? The answer is 100% absolutely yes. Okay? What we find is that the highest beach bacteria concentrations occur on a spring high tide. Right? What happens is the rack line, we have all the plants. Okay? What are plants? Well, I talked about agar before. Okay? What is agar? It's basically ground up plant material. Okay? When we have rack that gets formed up at the highest levels, it's essentially serving as an incubator for these bacteria. And when the highest high tides come in, they're the ones that reach that and cause it to recede. So I think we looked at that. We looked at the tidal condition at like 56 beaches in California. Every single one of them, we found the highest concentration. Again, receding tides, same thing. Land-based runoff. When you're doing a thing where most of the water is the ocean water coming in, that's not where your problem occurs. You had a second question, I think, about industrial. Industrial is really a different problem. That really goes to a chemical contamination. Industrial doesn't really put out bacteria. It's really the fecal material that you're worried about. So there are groups that do look at that, but it's not part of the beach monitoring programs. So I think you were next.
So you mentioned that you have the portable machines and then track sourcing. Are the portable machines also doing the source tracking? Because I know you need different primers and probes.
Okay, so the answer is absolutely yes. We have purposely built this to be dual channel. So you can actually, when the water goes in, it will actually process for both the indicator, the Enterococcus indicator, and the human fecal marker. So you can run 2 at one time. And with exactly that in mind. If you're going to go track, you want both pieces of information.
You mentioned that the qPCR, the new genetic method, is cheaper. I find that
Right.
unbelievable. And next, you said you have 3 monitoring test stations. I assume Cabrillo is one. What would the other 2 be?
Okay, so let me take the first question. I said it was cheaper. I probably should have said it has the potential to be cheaper. Okay, so there's three pieces to the cost equation. The first is, what does it cost to set up the laboratory? You know, the capital cost. The capital cost of setting up those 14 laboratories averaged somewhere between $75,000 and $100,000. Okay, and if you have a method, if you have a lab already that does culture-based, boom, you got a huge expense. The second piece of the equation are the material costs. The material costs for doing one of the traditional culture-based methods on the order of about $10. Okay? The cost of materials for doing the qPCR right now is probably more like $25, but that's because the industry hasn't truly developed all the way. We anticipate it's going to get down to about $5. Okay? The biggest difference is in the people time. And when you're doing the culture-based method, a lot of work to set it up and a lot of work at the back end to count it. When you're doing this more automated stuff, a lot less people time. So right now, I wouldn't call it cheaper, but as it gets developed over time, it will be cheaper.
And the three?
Oh, I'm sorry. Okay, so I said the 3 sites where we had that 55-gallon drum thing, Cabrillo was not one of those. Okay, we did one in San Diego at Ocean Beach Pier. We did one in Santa Cruz at the Santa Cruz Pier, because our partner was Stanford and they worked up there. And the third one, I'm sorry, I'll have to look it up. Not here. No, we did all those on piers. We did those at places that we needed a power source. We needed a pier where it was easy. What we were doing is we were mounting the device and then using a tube to pump the water up. It's not, here you don't really have a pier circumstance, so we'd have to find a way to put it on mooring. We're not at that level yet. So, Alan.
So why is the secondary treated sewage higher?
You know, I don't know the answer to that. I'll give you speculation. Speculation is that the ratio of pathogens to indicators changes. So what we do is we're real good when we treat our sewage. What we do is we chlorinate it, okay? And the chlorine is very effective at killing off the bacterial indicators, less effective at killing off the pathogens. That doesn't mean it doesn't kill pathogens, but it starts to change the ratio a little bit. That would be my speculation. So.
I'm curious about, so I've worked here at Cabrillo through the course of the sand replenishment or the replacement and then the introduction of the bird excluders. And I know, I love that you're able to detect what type of bacteria it is, but we definitely saw a decrease in the birds, especially right out here in the inner beach, after the implementation of the bird excluders. So do you have some speculation on why the bacteria seems to still be bird bacteria? I mean, if we're not seeing the number of birds, is that right in the water and that's being washed up in the rack? Or? Right,
so all kinds of reasons. I'll speculate for you that, again, speculation, that you saw how good the correlation was in the sand. And so what happens is sometimes the fecal material is in the sand and that provides a source for it to continue coming. So, right. Well, and again, you also see that your Enococcus numbers aren't as high as they used to be. So I think what happened is the numbers gone down, but I don't think you haven't got rid of all the birds for darn sure. So.
Steve, I have one quick question for you. I noticed your list of animals, you had cows and you had humans and, and others. I noticed there were 3 missing that I feel like are pretty strong on our beach. Raccoons, possums, and cats.
Okay. So maybe I don't want to swim at your beach anymore. So the answer to that is it's almost like, you know, the medical industry. You know, they want to put their dollars into the things that have the most. Raccoons probably is an oddity at most beaches. And so I don't know of any researcher who has developed a marker for raccoons. I think it could be easily done. It's just, what you need to do is you just need to get a whole bunch of raccoon poop and start to sequence it and figure out, what are the markers that work well with that. I just don't know any researcher who's done it. Cats is one that several people have brought up. I think there are some researchers working on that. It's just, what I did, because I wanted to save time for questions, I didn't go into more details about the evaluation steps that we did to prove that these methods worked. And when we were doing those evaluations, nobody had yet developed a cat marker. That would be a very logical one to do. Raccoons, I haven't heard of anybody who's working on it, but it could be done. Yes?
Are you doing any testing on Catalina, and because it's out in the ocean, if you are, is it less polluted out there?
Okay, we've done a lot of testing in Catalina. Catalina is a very interesting story. So we did one of our epidemiology studies out of Catalina. And you talk about it being out in the ocean. So I'm going to contrast Catalina as having two Catalinas. Okay? So if you've been to Catalina, you know there's a big enclosure, basically. And then there's the main beach along the kind of city. That has very limited circulation. Okay? Then you've got Descanso Beach. If you've been out if you kind of know where the dive park is, and then you go past the dive park, I guess it'd be northwest of the dive park. We never find any bacteria at Descanso Beach. Okay? You're going to Catalina, go to Descanso Beach. Okay? When we did the epidemiology study, one of the things that you all as a community have done is so completely screw up our scientists because you clean these beaches. We don't have enough dirty beaches to do studies anymore. Fortunately, Catalina was one of those. Okay? Catalina has historically, it's actually getting much, much better now, in part because of some of the studies that we did. But Catalina has historically a cast iron sewage system. Okay? And what do you flush your toilets with in Catalina? Is it fresh water? It's seawater. So what happens to cast iron when it gets 100 years of being flushed with seawater? It corrodes. And so what we found, very interesting patterns out of Catalina. What we found when we did epidemiology studies, first off, we found that people were getting ill, very high concentrations of human marker, and very cyclical. At high tide, we would find very little. At low tide, we'd find an incredible amount. Well, because what happens is that you've got this leaking stuff coming through the groundwater, and at high tide, you get essentially enough back pressure that the stuff doesn't drain from the land into the ocean or into the beaches nearby. But when it gets to be really low tide, there's really essentially nothing to push it back, and you get this stuff draining, and you have a lot of that human fecal material that's part. Again, I don't want to make it sound like it's Armageddon, because a lot of that is being filtered through the sand. And what do we use in a lot of cases of treatment plants? Sand filters. So it's not like it's astronomical, but we see a real clear pattern. And when we do the epidemiology studies, the correlation, the people who swim at low tide have a much higher chance of getting ill than the people who swim at high tide. And unfortunately, they're screwing that beach up by putting all new liners and the things. We can't use it for study anymore.
Anybody have that last question before we got through all our questions? Excellent. Steve, thank you so much.